Click the bell icon and subscribe to our channel so that you will not miss any classes. Hello students. In the previous class we have discussed the structure of megasporogenesis and we understood the structure of megasporogenesis in detail. As we discussed, megasporogenesis is covered by integuments. Generally, there are two integuments called the outer integument and the inner integument. That means normally in flowering plants, that is in angiosperms, there is presence of two integuments. But this condition will varies in different types of plants. Let us understood them. It is from competitive point of view, it is very important. In some plants, the ovule is said to be unitegmic, which means it consists of only one integument. Example, in case of higher dicots and gymnosperms. Ovule is said to be bitegmic. It is in case of bitegmic means it consists of two integuments. As we discussed, it is in case of monocots and primitive dicots. Dicots, generally speaking, in case of angiosperms or flowering plants. And ovule is said to be tritegmic, which consists of three integuments. Example, asphodelus. Ovule is said to be ategmic also. Ategmic means it do not contain integuments or else without integuments. Example for this is Santalum, Loranthus are the two examples of plants which bear the ovule without the integuments. So these points are said to be very important from competitive point of view along with the structure of ovule. Now let us understood in detail. We have understood already the structure of structure of megasporogenesis or the ovule, inverted ovule or the anatrophous ovule in the previous video. Now let us continue the next part of this, that is megasporogenesis. Megasporogenesis is the process of formation of megaspores from megaspore mother cell. Megaspore mother cell, otherwise it can be called MMC in short. Now to understand this process, that is where the megaspore is formed and how megaspore mother cells are formed. To understand this, let's once again write the structure of anatrophous ovule. This is anatrophous ovule or the inverted ovule, which consists of two integuments in it. This is one outer integument and inner integument. This is outer integument and this is inner integument. Inside this, there is presence of a nutritive tissue. As we know, the nutritive tissue present is the nucleus. And this end where the integuments incompletely cover the nucleus is called chalaza, and opposite to the chalaza is called micropyle. I am writing in short form. C refers to chalazal end and M refers to micropylar end. Now, one cell present towards the micropylar end, let us say this cell, and This cell is preferably the hypodermal cell. One hypodermal cell in the ovule where towards micropylar end. This point you have to remember. Hypodermal cell, this also you have to remember. It's not epidermal cell or nor integumental cell. It is the hypodermal nuclear cell. One hypodermal nuclear cell will become a microspore mother cell, and this is also referred as archisporium. In case of gymnosperm, this archisporium itself will develop into gametophyte. That is the different case. But here, in case of flowering plants, the microspore mother cell or the archisporium will divide mitotically and forms the megaspores. So let us write now. This is one megaspore mother cell. This is megaspore mother cell, MMC. Remember, this megaspore mother cell is having a dense cytoplasm and a prominent nucleus. And remember, this is diploid in condition. So these points you need to remember. Anatrophous ovule towards the micropylar end. One of the hypodermal cell will become megaspore mother cell. Otherwise, it is also called archisporium. This megaspore mother cell have a diploid nucleus, that is prominent one, and a dense cytoplasm. From this, there is development of megaspores. How they develop? I'll write here. This is microspore mother cell. For convenience, you need to remember the poles. This is chalazal end and this is micropylar end. This is megaspore mother cell where it is present. It is present towards the micropyle. This megaspore mother cell will undergoes meiosis. As we know, meiosis will produce us 4 haploid cells. 4 haploid cells are formed, and these 4 haploid cells are called now megaspores. And this process of formation of megaspores from megaspore mother cell, we have discussed already. The process is called megasporogenesis. This is 1 mark question. If they ask what is megasporogenesis, you can write the process of formation of megaspores from megaspore mother cell. And we need to remember, megaspore mother cell is diploid and megaspores are haploid. Now total how many megaspores are formed? 4 megaspores are formed, and this is called megaspore tetrad. Tetra means 4. Now important thing we need to remember is 3 megaspores present towards the chalaza will degenerate. That means they do not converted into anything. Degenerate. And 1 megaspore present towards the micropyle will become a functional megaspore. Functional megaspore. That means from this only there is development of embryo sac. That is, I'll write here now. This is functional megaspore. Same I am writing here. Remember, all the process what I am explaining, they are taking place inside the ovule. The functional megaspore, as I said, it also contain a prominent nucleus and a dense cytoplasm. This will develops into the functional megaspore will develops into female gametophyte. Gametophyte, or the other name for female gametophyte is embryo sac. And this process of formation of female gametophyte or embryo sac from the functional megaspore is called megagametogenesis. So remember, what is megasporogenesis? Process of formation of megaspore from megaspore mother cell. Now, from functional megaspore, process of formation of female gametophyte or embryo sac is called megagametogenesis. Okay. Now, how this process will take place? Now, I'll write here. This is functional megaspore with a prominent nucleus. Functional megaspore. The nucleus of functional megaspore will divide, and also the cell will enlarge in its size. It will divide by free nuclear division. It will divide by free nuclear division. One free nuclear division, that means only nucleus divide and there is no formation of cell wall surrounding the nucleus. Now after one division, mitotic division, there is formation of two nucleus. These two nucleus will move towards the opposite pole, one towards chalaza, one towards micropyle, and again they divide successively by two more divisions. Two more divisions will lead to formation of 4 nucleus at each pole. That means totally there is formation of 8 nucleus. After how many divisions? 3 free nuclear divisions. Free nuclear means you have to understood there is only division of nucleus and there is no formation of cell wall surrounding the formed nucleus. Now, one nucleus from each pole. So now remember, one nucleus from each pole will move towards the center and 3 nucleus will remain at chalaza, 3 will remain at micropyle. Later, there is formation of cell wall surrounding the 6 cells, which means 3 cells present towards the chalaza are surrounded or formed with the cell wall, and these 3 cells are called antipodals. And remaining 3 cells present towards micropyle are also surrounded by the cell wall, and one large cell is called egg cell and 2 pillar-like cells are present and they are called synergids. And synergids are having a thickenings, finger-like projections we can call them, and these finger-like projections are called filiform apparatus. So now there is remain of a large cell at the center, and this large cell at the center contain 2 nucleus in it, and these 2 nucleus, since they are from pole, one from chalaza, one from micropyle, they are called polar nuclei, and the cell which contain polar nuclei is called the central cell. Okay, so this is a complete embryo sac or the female gametophyte. We are calling it as female gametophyte because this entire structure is formed to carry the female gamete, that is egg cell. That's why this structure is called female gametophyte. As we studied in microspore or pollen grain. Pollen grain is called male gametophyte because it carries 2 male gametes in it. That's why it is called male gametophyte. Since this structure, that is embryo sac carrying 1 egg cell, it is referred as female gametophyte. Now, remaining things, some few more things we need to understand regarding the development of embryo sac. During the course of this study, we have understood that embryo sac is developed from a single functional megaspore. Single functional megaspore. So, this development is called monosporic development. And second thing, as we studied the structure of embryo sac is containing different types of cells and number, different types and number. So see here, let us count the number of cells in it. Here it contains 3 antipodals, 2 synergids, 1 egg cell and 1 central cell, which means how many cells are present in it? 7 cells. And all the 7 cells are of different type. So this is called polygonum. Polygonum type, which is made up of different types and number of cells. And generally the embryo sac of angiosperms is called 7-celled and 8 nucleated. 7-celled and 8 nucleated. These points you need to remember. Development of embryo sac is monosporic because it develops from a single functional megaspore. Secondly, it is polygonum type because it is made up of different types and number of cells. And embryo sac is said to be 7-celled and 8 nucleated. So that's all about the development of embryo sac. So embryo sac development from the single functional megaspore is called megagametogenesis, and development of megaspore from megaspore mother cell is called megasporogenesis. Okay, so in next class we shall take up the process of transfer of gametes. So in event of sexual reproduction, we have studied that there are 3 events: pre-fertilization, fertilization and post-fertilization. Pre-fertilization events are 2. They are formation of gametes and transfer of gametes. Here we have understood development of microspore and development of megaspore, or development of male gametophyte and development of female gametophyte. Respectively, in male gametophyte there is formation of male gamete. In female gametophyte there is formation of female gamete. To get fertilized, these two should be brought together, and that process we call it as gamete transfer. In case of angiosperms, gamete transfer will take place through the process called pollination. That we shall take up in the next class. Okay, thank you.